So good morning. You are very welcome to the second day for our symposium. Yesterday it was a full day with a lot of plenaries and oral presentation, poster presentation. And now also we have a rather busy day with plenaries and all the other sections. So it is my pleasure to invite Professor Milen GEORGIEV to present his plenary. But first of all, I ask Professor Dr. Radu Fieres to introduce him. Thank you.
Thank you very much. Good morning, everyone. So, as you see, Professor Milen GEORGIEV is the head of Metabolomics Laboratory, the Institute of Microbiology from the Bulgarian Academy of Sciences. Milen has over 20 years of experience in natural product fields, has published over 180 papers in very good journals. We all know his work. Has delivered over 60 invited lectures in the 20 different countries, one of them being also Romania last edition. Milen holds several grants from a National Science Fund of Bulgaria, Romania, and framework programs of the European Union, including an H2020 Plant Assist project with a very good budget, over 30 million euros, on which he built an impressive new institute practically. For the last 3 consecutive years, he is listed among the top 2% of the Stanford University worldwide citation rate ranking, and the recipient of a Pythagoras Award for Outstanding Scientist by the Bulgarian Ministry of Education and Science for 3 times, making him the only researcher in Bulgaria to have won it 3 times. Milen is an editor, associate editor, and editorial board member of a dozen journals in biotechnology and natural product fields, including phytomedicine, food and chemical toxicology, phytochemistry, biotechnology letters, Chinese medicine, food frontiers, International Journal of Molecular Sciences, Frontiers in Pharmacology, and many others. Serving as a chairman, he has established the International Conference on Natural Product Utilization from Plants to Pharmacy Shelf, which has 4 issues so far. The latest ICNPU edition attracted the attention of more than 330 participants from 50 different countries, and we are expecting the 5th edition as soon as possible. His current research focuses on the biosynthesis of fine molecules and development of biotech tools for their sustainable mass production, metabolomics and comprehensive metabolite profiling, and molecular pharmacology, with a particular focus on obesity, photoaging, and longevity. Milen, please you have the floor.
Can I have the pointer? Okay, so thank you, Radu, for the nice introduction, and good morning, everyone. First of all, I would like to thank the organizers for the invitation to come here. So I've been maybe hundreds of times in Bucharest, but I've never been in this institute, so it's my first time, and I like the space, and yeah. So yeah, I have a Romanian CNP, but I cannot speak Romanian, so I assume most of the people in the audience don't speak Bulgarian, so I have to make it in English. What I'm going to speak, I choose to speak about obesity. This is one of the domains which we are trying to cover in my lab. So it's my primary affiliation is Bulgarian Academy of Sciences. My lab is, we name it Metabolomics, and we, as Radu mentioned, we establish a Center of Excellence. We name it Plant Systems Biology and Biotechnology. So we established it already back in 2015, and we managed to get a good support from European Commission under Horizon 2020, Bulgarian government, and Plovdiv municipality accounting in total of more than 35 million US dollars or euro, because they are equal now. So this is the campus. We just finished in the construction in May, and the structure of the departments, some papers, some nice papers we got already till now in 2020. But what makes us happy is that we managed to buy some nice pieces of equipment. So this GCMS is quite traditional, but the LCMS is very nice. So it is tough, it is with ion mobility, and it has this RAMS device which is called iKnife. So basically, when you work with plants, what we do, we produce extracts, and the extracts sometimes with the extracts you produce artifacts. So if you want to explain the native states within the cells, eventually you need to work as it is in the intact mode. Of course, you can always try to do some magic angle spin in NMR, but with the mass spectrometry, with the iKnife, it's also possible. So this was originally developed for cancer and patented for cancer from waters, but we plan to use it on natural products field as well. So it's a quite unique design, and unfortunately it's not that unique because in Cluj-Napoca they have already one, but this is eventually the second in the region. So we plan to do quite a lot of work because it's a UPLC, so methods are 10 minutes, so you can run a lot of samples per day. So this is a good machine for the collaboration, I believe. Okay, so plants, we use plants for food and feed and for many purposes, but plants also accumulate small portion of compounds which are called secondary metabolites. Right now we have a discussion whether we should call them secondary metabolites or specialized metabolites, but in any case, many of them have shown beneficial properties for the human health. So here on the slide, I put some of the blockbusters, and in the middle are paclitaxel, which is used to treat different types of cancer, and artemisinin, whose discovery was awarded with the Nobel Prize a couple of years ago to the Chinese scientist Yu Tu. So when they announced the Nobel Prize award, so we from the Phytomedicine Journal, we were very interested to make an interview with her. She's not speaking English, but we sent the questions, and we asked how she made the discovery, and she replied in a way like, it's not any kind of my discovery, this is because of the chairman Mao. So we decided not to put this interview in the journal, but I think Romanian people or a certain generation understand this situation, you know. Okay, so in addition to because all of these are pure compounds, in addition, so 80% of it is estimated by the WHO that 80% of the human population still rely on herbal medicines for their primary health care needs. And I think this is quite representative for the region, although unfortunately this is changing. So what we do in my lab, we use medicinal and aromatic plants from all around the globe, not only from European flora. This one on the top is a very beautiful plant. It's called Devil's Claw or Harpagophytum procumbens, and it grows only in Kalahari Desert region in Africa. We do isolation and structure elucidation. Mostly we used we used NMR. Now we are implementing also mass spectrometry, and with the extracts and pure compounds, we have several in vivo and in vitro assays. So right now we are interested in obesity, longevity, photoaging. This is the aging of the skin when exposed to UV light and the blue light, and also psoriasis. This is something interesting. We have a product and we are trying to push a little bit more because psoriasis is a sort of quite unpleasant disease. So as I mentioned, I will speak about obesity, and longevity is a really hot topic nowadays. So in Romania, I was last month in Iași, and many people say, we have the key to longevity in Romania, just drinking palinka. You know, so I'm not quite sure whether it's true. So I cannot say this recipe is verified, but many people in Romania believe the key to longevity is drinking palinka every day. You know, okay. Once you have a lead compound, there are two possibilities. So to go through the preclinical and clinical studies to end up with a product. It happened to me only once in my life so far. In most cases, our compounds are going to the rubbish bin. So this rubbish bin is supposed to be full of garbage, but in our case, it's full of natural compounds. This place here is a space for lead finding acceleration, I believe. So what we do, we try to correlate some specific signals from the NMR with some particular biological activity. The idea is just to have a sort of fast screening. You know, proton NMR takes 5 minutes. And if you are, for instance, seeking for some compounds with anti-cancer activity, you might need to go to 72 hours. So the idea is you run a proton NMR, you check for some particular signals. If you see them, you go to more sophisticated assays. If you don't see, then the compounds go to the rubbish bin, and then you continue with the next one. Theoretically, it's very easy. Practically not, unfortunately. Okay, so obesity is a real pandemic. So it has escalated more than 3 times in the past 40 years. So here on the top, you can see the map with this map is representing the obesity with body mass index of the people with body mass index above 30. So as with the dark pink color are the countries which are mostly affected by obesity. So of course, States, Canada. So Romania is here. So the situation is not. So this is Black Sea, and Romania is here. So the situation is not that terrible, but the projection is that this color here for Romania, Bulgaria, and most of the East European countries is going to change into a dark pink. So the number of the people who will have obesity, who will develop obesity, will increase, continuously increase in future. So the situation now, how to treat obesity. So I divided this based on the body mass index. Body mass index has nothing to do with mass spectrometry or NMR, but it is quite useful and handy when you go to your general practitioner to measure your body mass index. So basically, the body mass index between 19 and 25 is considered as the perfect one. And if you have such body mass index, you don't need to listen to this talk, you know. But for the people with 25 and 30, this is the overweight stage. And actually, the recommendation is, the first recommendation is changes in the lifestyle. Then for the obesity stage with the body mass index between 30 and 40 is eventually the drug therapy a solution. And for the excessive obesity with body mass index above 40, eventually the bariatric surgery is a solution. So as you can see, neither of these approaches is enough effective. So in other words, there is a need to develop a new effective and eventually safe therapy treatment-wise. So this is a summary of the research direction lines which we follow in the obesity research. So basically, there are two processes: accumulation of fat into the adipocytes. The adipocytes are our fat cells. So one part is accumulation of the fat, which is called adipogenesis, and one would eventually need to abstract this process. And the second is lipolysis. Lipolysis is the release of the fat out of the cells, and the induction of the lipolysis is one of the solutions to cope with obesity. So browning is very interesting. WAT stands for white adipose tissue. So making this white adipose tissue brown is eventually a solution, because at the children stage, we take quite a lot of in excessive amount of energy and carbohydrates, but the children are mostly not obese because most of the fat tissue is brown, which is easily burned. And one of the solutions to cope with obesity is to try to adjust the genetic program of the white adipose tissue to turn into brown. Another solution is, or a direction of research is to cope with the inflammation, because with the obesity, obese people develop low-grade chronic inflammation, which is already known that at certain point, this inflammation, continuous inflammation, is bringing neoplasms and development of different types of cancer. So here on the slide, you can see how the adipocytes are formed. And to cut the long story short, I will show you some data from these transcriptional factors, PPR gamma and CBP alpha, which are differentiation markers for the development of major adipocytes. So what we do, when we started working on this domain a couple of years ago, we decided to step on the ethnopharmacology based. So many people say that ethnopharmacology is a sort of not that well scientifically sound and not quite fashionable nowadays, but we still need to keep in mind that ethnopharmacology is based on human studies. Now we start with the cell cultures, we go to some animal models, and if you have a lead compound, you can eventually go to clinical studies. But all of the ethnopharmacology knowledge is already on humans. So with our modern tools and techniques, we can prove or disprove. So if you prove that particular plant or bioactive compounds from the plant works, it's nice, you might end up with a product. But if you say that it's not working, of course, you are not going to publish in a good journal eventually, because there is no journal of negative results. But still, this is good for the society, because, for instance, in cancer field, people are desperate and they want to buy whatever stuff for their relatives. Sometimes it's quite expensive, and if you can say, hey guys, it's not working, so this is also good, because they are not going to waste their money. Okay, so as I mentioned, we generate extracts, pure compounds. We do NMR at every stage, and we use HGBS adipocytes. So HGBS stands for the Simpson-Guabi-Bemmel syndrome. This is a rare genetic disease, fortunately, and people who are suffering, individuals who are suffering from this genetic disease, they tend to develop obesity and cancer. So we treat adipocytes with extracts and pure compounds. We isolate RNA and do RT-qPCR, Western blotting to check the expression of some proteins, and also metabolomics. And to extend from the cell cultures, we also utilize recently in my lab the C. elegans model. So we publish a couple of papers on some plants like Ziziphus, Alchemilla, and Unonis. Thanks to the support from European Union, all of these are open access. So you, for the sake of time, I can focus only on one of them, but if you want to read them, you can freely download them. Okay, so what we did, we took Ziziphus jujuba, and we produced an extract. So searching for a natural compound with particular biological activity is like searching for a needle in a haystack. So at the beginning, we started with the molecular docking studies, and from them we figure out that 3 molecules from the extract in Ziziphus, present in Ziziphus jujuba, like apigenin, betulinic acid, and muscinic acid, are theoretically binding to these proteins which we are interested in. So here you can see the data. This is the inhibition of adipogenesis. As I mentioned, adipogenesis is the formation of the fat into the cells, and at the bottom is adipolysis, release of this fat. So we didn't observe any significant activity to induction of adipolysis, but if you look here with respect to the inhibition of adipogenesis, the extract and the pure betulinic acid appeared quite active. So this is the highest concentration of the extract. You can see what's going on with the adipocytes, and here is the highest concentration of betulinic acid. So then we check for some transcriptional factors or major players in adipogenesis formation. As I mentioned, PPR gamma and CBP alpha are 2 transcriptional factors which are taking part in the adipocytes later stages of formation. ACC stands for the acetyl coenzyme A carboxylase, which is an enzyme which takes part in the biogenesis of fatty acid. So again, we figure out that extract as well as betulinic acid appeared quite active. So here at the bottom panel is the situation at the translational level. So these are the proteins. This is how the Western blot looks like: PPR gamma, CBP alpha, and adiponectin. So we were also interested to try to explain how the activity of Ziziphus and the pure compounds is going through. So we check for the PI3K act. PI3K act stands for these are kinases. PI3K is phosphoinositide free kinase, and ACT is protein kinase B. So this is a highly conserved pathway which is taking part in the energy balance, and it's not only in obesity, but it's already shown to take part in different types of cancer development. So we figure out that the extract again and pure betulinic acid appear quite active. So to cut the long story short, this is the eventual mechanism of action of the Ziziphus jujuba and the pure compound. So when the activity is triggered from the insulin receptor, there is a cascade of reaction which are led by PI3K act. As a result, some transcriptional factors in the nucleus are overexpressed, and finally increased lipid accumulation, ACC and adiponectin is observed. So when we treated the adipocytes with the extract and the pure compounds, we managed to disrupt this process at different stages. So as I mentioned, one of the strategies to cope with obesity is to cope with the low-grade chronic inflammation. So here is one paper we published last year. This is rosmarinic acid, natural compound, very beautiful compound. It's a dimer of caffeic acid and 3,4-dihydroxyphenyl lactic acid. So rosmarinic acid is known to have anti-inflammatory properties. But what is already known for many natural compounds is that this activity is influenced is different in different types of plant tissues and organs. So we check in the adipocytes and we figure out that rosmarinic acid is inhibiting adipogenic genes and proteins at transcriptional as well as at translational level. So at the next stage, we decided to combine two molecules: caffeic acid, this is actually half rosmarinic acid, and chlorogenic acid. And when we combine them at the concentration of 50 micromoles here, you can see the highest concentration. We observe a very beautiful stimulation of adipolysis, which is actually a release of the fat which is already accumulated. So this is the mechanism of action. So I think I'm running out of time, so I can just briefly go through this, but we figure out that the major player in this case is AMPK, which is activating and inhibiting several processes, but UCP1 is a Brownian gene, which is quite interesting for us, and we are following it. So C. elegans, all of the data which I showed you is based on the cell culture. Cell cultures are very beautiful when you want to do a preliminary screening, but if you want to go a step further, you need eventually organism level. So nowadays in European Union, it becomes very difficult to work with rodent models. I think the situation in Romania is also similar, or at least is going to change, unfortunately, into bad direction. So that's why we decided to use C. elegans, because there are no ethical restrictions in this direction. So what is interesting is that the genome sequence is sequenced. There is a relatively high homology with the humans, and these worms are a very nice model to study obesity and longevity. So here you can see this is the confocal microscope images control, 2% of glucose and 10% of glucose. So you can see how we make the worms fat. We use 2% of glucose, which is a gold standard nowadays for this type of research. So before starting, you need to check that the compound which you are going to use is not interfering with the different stages of development of the C. elegans, like growth size, daily progeny production, bending movements, and so on. And here you can see the data. This is when what's going on with the worms when they were treated with the betulinic acid. This compound at the bottom has nothing to do with the betulinic acid, as you can see from the chemical point of view, but this is orlistat. It is already approved drug for treatment of obesity. So we figure out that both betulinic acid and orlistat appear quite active. So then we said, okay, can we eventually combine them to make a hybrid combination, which is quite interesting and quite frequently used in cancer field. So this is the data. This is orlistat, then is betulinic. When we combine them, we observe completely, this is the lowest value means better activity. So the best was observed for the orlistat, then betulinic acid, and when we combine them, we didn't see that activity which we wanted to see. But anyway, it's a good approach to go further. So this is the expression profiles of the genes which we are following. We divided them in those who takes part in the lipogenesis, the formation of the fat, lipolysis, and these here three in the middle are taking part in both lipolysis and lipogenesis. So to cut the long story short, we figure out that some genes which are particularly interesting for us are down-regulated when treated with the betulinic acid. And in particular, this one, NHR49, is a nuclear hormone receptor 49, which is a functional homolog of the human PPRs. So we also check for the expression of some microRNA in the worm. So we pick up all microRNAs which were known to take part in obesity development in the human body. So then we check for their orthologs in the C. elegans, and we figure out that all these here were down-regulated, while this one was upregulated when treated with orlistat. So this is a summary of the work which was just published this month. So basically, we figure out that the activity of betulinic acid goes through the down-regulation at the highest concentration, upregulation at the lowest concentration of NHR49, which is a homolog of the PPRs, and it's also essential for the lipid metabolism and beta oxidation. So the last topic is longevity. So here you can see this is the projection from the World Health Organization, what is going to be by 2050, which is not that far from now. So this is the percentage of the people above 60, at the age of 60 or above. So here you can see in the blue, in the dark blue, are the regions where 30% of the people of the population will be above 60. So again, see, now Romania is the same situation as most of European countries. So our population is getting older. So in a linear age, this is our life. So I think the most enjoyable period is around here, so-called reproductive period. But as we are about to hit, so I will hit this in few years. Radu is a little bit younger, but you will be also soon here. So with after 50, you can see what we can expect: aging-related diseases, so cancer, stroke, Alzheimer's, and so on. So we build a hypothesis that health and longevity can be affected by slowing aging. So actually, as I mentioned, longevity is a really hot topic in States. Many people invest quite a lot of money in research in longevity and in startups on this direction. Also, 2 months ago in August, Saudi Arabia announced that they will open a new institute focused exclusively on longevity, and they will put $1 billion on this. So European Union is a little bit behind, but I believe this is going to change in the very near future. So, and I believe that the natural compounds, they have quite a lot to do into this direction. So we are pushing quite a lot. And if you have a compound which you believe that can affect the longevity, so we'll be happy to check in our lab. So many people say that this is a fiction, but I would just like to remind that 100 years ago, the average life of the human population was 50 years. So now at 2020, it was 80. So only in 120 years, we managed to extend by 60%. So just imagine if we manage to extend with another 60, what it will be. Of course, then we have to work until 100 years till retirement. But anyway, you know, in science, we never retire. So, okay, as Radu mentioned, we are organizing a meeting. This is the last one just before pandemic. As you can see, many happy people. So Radu is somewhere here also. And the next one is planned again at Black Sea coast for the end of May. And these are the people who contributed to this work. This is my lab. So as you can see, we have nearly perfect gender balance, because for all projects we have to write about gender balance. So we already have this gender balance, sorry. And at last but not least, thank you for your attention.
Thank you very much, Milen. If you have any questions from the room, please.
Thank you. Congratulations for your whole work. I want to ask you, how fast do you think that is cell lipolysis? If it's too fast, I'm think that it will affect the level of let's say greasy triglycerides and cholesterol in lymph and in blood. And do you think that it will be very fast or not?
Yeah, well, that's a quite interesting question, because of course, when you have a fat which is accumulated into the adipocytes and this fat is released, this is activating a lot of enzymes and parts of our biochemical machinery. But you know, this is just one of the points. So from all these possibilities to cope with obesity, I cannot say there is one which is the best. I believe we need to target at least several of them if we want to have an effect, you know. So adipolysis is one of them, which is eventually a solution when people already develop obesity, you know. So at this stage, all of the human systems or the individual who is suffering are somehow disrupted, you know. So and the fat cells, the adipocytes, they are made in a way like, you know, up to the middle of the last century, human population was living in absence of excessive amount of food, you know. So we used to accumulate, like animals in the nature. We used to accumulate, to take everything from the food and eventually to store, because we don't know where will be the next time when you will have access to the food. Well, now we have an excessive amount of food, and of course carbohydrates, which to me are the or the white and beige carbohydrates are the biggest evil. But the idea is that naturally we can get rid of this energy very easily when you need to escape. So I think this is not the main one. Of course, this is going to increase several enzymes, but just imagine if you have a severe obesity, you don't have another opportunity. But I think the most effective strategy would be to cope with several of the processes rather than just focusing on lipolysis. We never found a molecule, even synthetic ones, which we screen, which is extremely potent. Okay, combination of caffeic acid and chlorogenic acid is stimulating adipolysis, but this is not so much drastic in a way like saying, okay, you take it, then the adipocytes are just releasing everything, and at the day after you are slim.
I'm asking because recently we learned about a case here in Romania, a young boy, who lost half of his mass. So from 100 kilos to 50 or less than 50 in a very short time, and he's now in a very bad situation with his health. And this is idea to be very carefully to balance and to dose to make a right dose of this. I don't know what pills probably some chemical synthesized, not natural.
Yeah, you know, this is quite valid what you say. What is a big problem with this industry is that it is a sort of dirty. Most of these products are food additives, so they are not drugs. They are not registered as drugs. So they go through a very soft procedure for registration, and we have now check-in by ANMR what is the content of many of these, because in Bulgaria there is now a product which is very effective. Obviously people lose weight, and one of my assistants used it, and when I asked him how did he feel, he described the side effects of sibutramine. You know, and this is a big problem. Many of these products are adulterated with sibutramine, which is banned in European Union for many years. Sibutramine works in obesity, but it also brings severe problems with the heart, which can be life-threatening. You know? So it could be such a situation which is just a guess. But we check the market. That's why I say it's a dirty. There is a huge percentage of adulteration with sibutramine. It's not written on the label. It says natural, something like this, and then you get you lose weight, but you also get some heart disease or whatever stuff, which is extremely unpleasant and can be left.
Yeah, any more questions, please?
Congratulations, very nice presentations. Presentation. I want to ask you, because you address the problem to person having already the problem with obesity, but do you think to discover something to address to the young people, or because the obesity is not like getting the problem during the life, it's a genetic one. So maybe to come with a product early before developing obesity, and to, yes, a product to stop or to inhibit, I don't know. Because all the people are thinking, okay, you are obese, you must take this or to do this, but the problem is not there. It's to make an awareness and to come before developing the biggest problem, obesity. And unfortunately, starting with young people from very early age.
Yeah, that's a very good question. So what we know from cancer is that between 5 and 10% of the cases are genetically related. So I believe in obesity is more or less so, which means 90% to 95% is from the lifestyle. You know, so of course, these people who have a genetic predisposition to develop obesity, then I think it will be very, very difficult, because these cells which we used, they are from the patients who are genetically predisposed to develop obesity, and they are not as sensitive as cells from the patients who don't suffer from such disease. So I think this is quite complicated, and but yeah, you are right. So it's better first to aware people before developing obesity, rather than, which is maybe for us is valid, not for the pharma industry, because pharma industry wants patience as long as possible, you know. So these are different stages, but yeah, you are absolutely right. So if you can prevent them from, but we have a lot of knowledge nowadays for the healthy eating style and so on, but how many people are following this? You know? Yeah, that's.
Thank you. We have any more questions, or from the online? If not, I would like to thank Milen for his participation. We will surely come to the 5th edition of your conference. And I think the next year will be more of our colleagues coming also. And good luck, I don't know, making the 80s the new 20s. Okay, thank you very much, Milen.